Alright, this is a lecture on photosynthesis. This is actually one of two. There's going to be photosynthesis 1, which is today in this lecture. So we're looking at photosynthesis. The first thing to look at is the fact that we have a chemical equation that supports photosynthesis, and that is carbon dioxide, which you need 6 of those, plus 6 water in the presence of light energy will make glucose, C6H12O6, plus 6 oxygen. And when we talk about photosynthesis, we usually say that we're talking about autotrophs, but in this equation, you can see that we're looking at the presence of light. So we actually make that a little bit more complex now and say we're looking at photoautotrophs. And you think about the things that do photoautotrophs, a lot of times we obviously think of a tree or grass, some type of plant, maybe a vegetable or a fruit. Those are all photoautotrophs, but there's also some protists out there, there's some algae out there, there's some other organisms that are also doing photosynthesis. So keep that in mind as you're looking at photoautotrophs as well. So when we look at the presence of photosynthesis, what we find is that this takes place in the chloroplast of the plant cell. Now most of the time when you see a plant cell drawn in a book, or even if we've done it in class, you know that usually we include 1 or 2 chloroplasts. That's one of the things that you include in a plant cell. And that is correct, but when you actually look at a chloroplast in a cell, a mesophyll cell, let's say, that is a cell in the leaf that is primarily responsible for photosynthesis, you'll actually find 30 to 40 of these chloroplasts in that. When you do the math, that equals about 500,000 chloroplasts per millimeter squared section of the leaf. That is extremely small and extremely large number of chloroplasts in there, but it's because chloroplasts are so essential to doing this process of photosynthesis. So I want to identify a couple things about the chloroplast first that you can see here. The first is looking at the fact that it's surrounded by a double membrane. There are 2 membranes that go around it. That's important because that supports endosymbiotic theory that we've talked about earlier in class. The other is that it has a cytoplasm-like material in here that's the background information here, and that is called the stroma, and that's going to be key to where one of the parts of photosynthesis take place. You have these stacks of green discs in here, and when you look at all of them together, you're talking about the grana. But when you just talk about one of those stacks, you're talking about the granum. Now those little discs themselves are called thylakoids, and that is key to understand, because in this chloroplast, remember there's 30 to 40 chloroplasts in a mesophyll cell. There's tons of these thylakoid discs in there, and that is going to be where one of the primary steps of photosynthesis take place. So when you think about photosynthesis, we're going to look at it with just one thylakoid, but this is going on in all of those thylakoids, in all of those chloroplasts in that cell. So you get a lot of this happening at one time. So let's jump right into it then and look at the first step of photosynthesis, which is called the light-dependent reaction. It's obviously called that because it requires the addition of light energy. There's another step that we'll talk about later that doesn't. So when we look at the light dependent, the first is understanding, one, where we figured out what is being absorbed by light. And that comes to an experiment done by Thomas Engelmann. Thomas Engelmann took a strip of photosynthetic algae and he put a layer of aerobic bacteria on this photosynthetic algae. And then he hit this algae with a prism, a light that was shining through a prism, so it gave all of the spectrum, the visible light spectrum, kind of like a rainbow, okay, from the blue all the way over to the reds, okay? And then he let that set. The photosynthetic algae is doing photosynthesis. One of the byproducts of photosynthesis is oxygen, which those aerobic bacteria need. So after he allows this to go on for a while, he comes back, he looks, and he's got a bunch of aerobic bacteria growing in the section that's being hit by blue and purple light, and a bunch over here that's being hit by the reds and the orange lights. So with that, the hypothesis that would come from that is that apparently more photosynthesis is being done in the visible light spectrum of the blues and the purples and the reds and the oranges. Now, with more technology and advancement, we do know that that is the case today. that green light is more reflected, and that's why they appear green, and the other lights are more absorbed. Now, what actually absorbs those are pigments, and there's 3 different pigments in photosynthesis that we look at: chlorophyll A, chlorophyll B, and the carotenoids. Chlorophyll A and B are the primary pigments, and those are the ones that give the leaf its green color by reflecting that green light. The carotenoids you see more in plants that have purple or red fruits, yellow fruits, orange fruits, as well as in deciduous forests when the leaves start to change colors. The chlorophyll A and B is what shuts down. The carotenoids are still going, and that's why you start to see some of those orange and reds and yellow leaves and that sort of thing, is it's reflecting those colors of light. Also, when you look at the chlorophyll molecule, many times we describe it as a green disc looking shape with a hydrocarbon tail, and that hydrocarbon tail is going to hold it in place. And in that green disc, right in the middle of that green disc, is a magnesium element. And that is important because magnesium is going to be what throws off its electrons, which is what causes this whole process to be possible, is the fact that magnesium is right there in the middle. So let's look at it like dependent reaction. So we've got a thylakoid, which has a membrane around it, just a normal phospholipid bilayer. Okay, and I've drawn that with 2 lines. There's proteins embedded in that membrane, just like we've talked about before with the cell membrane. But there's 2 important ones I want you to take note of. The first one is right here. This one is called photosystem 2. And if you'll see all these little green dots on the top of it, those are all chlorophyll molecules setting on that. This one here is called photosystem 1. It also has a bunch of chlorophyll dots on top of it because it also has chlorophyll on it. So we usually start over here with photosystem 2. So light energy is absorbed by the chlorophyll. That causes that magnesium to throw off its electron. As it throws off that electron, it's going to pass that electron down the electron transport chain, which is a series of proteins that pass that electron from one to the other, kind of like hot potato. And we've talked before about the fact that when electrons are passed, they put off energy, which is the whole point. So as that electron is being passed, it's releasing energy. This protein right here is called a proton pump, and it's going to harness that energy to push hydrogens or pump hydrogens or protons into the thylakoid. That creates a hydrogen concentration gradient, and we're going to come back to that in just a minute. So these electrons are passed one to the next. Now when it gets to here, another thing takes place. Light is being absorbed here. Magnesium in these chlorophyll are throwing electrons. Those electrons are picked up out here, and eventually those electrons are picked up by NADP+, which turns them into NADPH. Remember, NADPH is an electron carrier, and an electron carrier's job is to carry electrons, which is exactly what it means. I like to think of it as a dump truck, where those electrons are being put into the truck and driven to another point in the cell. And you're going to see where that point is later. So now we've got this hydrogen concentration gradient that's being increased. We've got a magnesium molecule that's missing an electron here that has to be taken care of. So water, what you see here, comes in and binds. This protein will split water, give the electrons back to magnesium, and release oxygen and hydrogens. So that also goes to the hydrogen concentration gradient that's being produced. So you've got a large amount of hydrogens in here that need to get out now. They want to go down their concentration gradient, passive transport, diffuse out of the thylakoid. They use this molecule right here, which is ATP synthase. ATP synthase is going to allow those hydrogens to diffuse out, and as it does, this ball down here on the bottom will spin. All right, it's a poor drawing, but it's the best I could do. This ball will spin, and as it spins, it's going to give off energy. That energy is going to be picked up by ADP and a phosphate and turned into ATP. And that's the energy molecule for cells. Now, what you don't want to get confused on here is whether photosynthesis is a primary mechanism for making ATPs. which is what a lot of students think because they see ATP right here. That is incorrect. ATP is not made in abundance in photosynthesis. ATP and NADPH are both going to be used completely up in the next step of photosynthesis.